प्रिय विद्यार्थियों, हमने प्लांट ब्रीडिंग के मेथड्स में प्रथम भाग में हमने मास सिलेक्शन, प्योर लाइन सिलेक्शन और प्लांट इंट्रोडक्शन एंड एक्लीमेटाइजेशन के बारे में अध्ययन किया था। अब हम इसके द्वितीय भाग के अंदर बची हुई विधियों और साथ में अलैंगिक जनन के द्वारा किस प्रकार से पौधों को बहुगुणित किया जा सकता है उसके बारे में भी हम अध्ययन करेंगे। सबसे पहले हम बात करते हैं संकरण की। द प्रोडक्शन ऑफ ए हाइब्रिड बाय क्रॉसिंग टू इंडिविजुअल्स ऑफ अनलाइक जेनेटिक कॉन्स्टिट्यूशन is known as hybridization. Hybridization is an important method of combining characters of different plants. Hybridization does not change genetic contents of an organism, but it produces new combination of genes. संकरण वह प्रक्रिया है जिसमें हम किन्हीं दो अलग जेनेटिक मेकअप वाले प्लांट्स को लेकर के एक संकर बनाते हैं और वो संकर कभी भी उसके जेनेटिक कंटेंट को परिवर्तित नहीं करता है, लेकिन वो नए कॉम्बिनेशंस का, नए संयोगों का निर्माण करता है। संकरण के उद्देश्य क्या है? पहला उद्देश्य टू आर्टिफिशियली क्रिएट ए वेरिएबल पॉपुलेशन फॉर द सिलेक्शन ऑफ टाइप्स विद डिजायरेबल combination of characters, to combine the desired characters into a single individual, and to exploit and utilize the hybrid varieties of different crops. So hybridization just gives us new variety with superior characters or the superior combination of genes to produce such individuals which are superior to the existing ones. The mutation breeding is another method to improve the crops. The aim so at accelerating the process of developing different traits for selection such as disease resistance, tolerance to harsh growing conditions, and other valuable agronomic traits. It does not involve gene modification, also known as gene splicing. Mutation breeding uses a plant on genetic resources, mimicking the process of spontaneous mutation that is underway in nature all the time. The basis of evolution, importantly, it broadens biodiversity. Mutation breeding is built on mutation induction and mutation detection. Mutation induction coupled with selection remains the cleanest and most inexpensive way to create varieties by changing single characters without affecting the overall phenotype. So म्यूटेशन ब्रीडिंग और जिसे हम प्रजनन का एक ऐसा तरीका है जिसमें जीन्स के अंदर परिवर्तन करके, जीन्स के अंदर कुछ चेंजेज करके जो है उसके अंदर उत्परिवर्तन करके हम एक नई वैरायटी का निर्माण करते हैं जो कि बहुत सारे प्लांट के जेनेटिक मेकअप को परिवर्तित ना करके कुछ जीनों में ही परिवर्तन करके और ऐसी सुपीरियर वैरायटी का निर्माण करता है जो कि अच्छी उपज और हमें एक अच्छी वैरायटी उपलब्ध कराता है। ब्रीडिंग मेथड्स इन क्रॉस पोलिनेटेड स्पीसीज, दैट इज मास सिलेक्शन, डेवलपमेंट ऑफ हाइब्रिड वैरायटीज एंड डेवलपमेंट ऑफ सिंथेटिक वैरायटीज। मास सिलेक्शन इन क्रॉस पोलिनेटेड स्पीसीज टेक्स द सेम फॉर्म as in self-pollinated species. Large number of superior appearing plants are selected and harvested in bulks, and then seed used to produce the next generation. Mass selection has proved to be very effective in improving qualitative characters and applied over many generations. It is the general method used by the pigeons that they select the superior variety of seeds, the more in weight, the more in size, then they are selected and kept for the next generation to be grown in the field. It is also capable of improving quantitative characters including yield. Despite the low heritability of such characters, mass selection has long been a major method of breeding cross-pollinated species, especially in the economically less important species. Hybrid varieties, the outstanding example of exploitation of hybrid vigor through the usage of F1 hybrid variety has been with the corn, maize. Production of a hybrid corn involves 3 steps: the selection of superior plants, selfing for several generations to produce a series of inbred lines, which, although different from each other, are each pure breeding and highly uniform, and the crossing selected inbred lines. So hybrid variety can be created using these three steps, and the hybrid obtained is very vigorous in the first generation. During the inbreeding process, the vigor of the line decreases drastically, usually to less than half that of field pollinated varieties. Vigor is restored. However, when any two unrelated inbred lines are crossed, and in some cases the F1 hybrids between inbred lines are much superior to open pollinated varieties. An important consequences of the homozygosity of the inbred lines is that the hybrid between any two inbreds will always be the same. Once the inbreds that give the best hybrid have been identified, any desired amount of hybrid seed can be produced. The synthetic variety is developed by intercrossing a number of genotypes of known superior combining ability, genotypes that are known to give superior hybrid performance when crossed in all combinations. By contrast, a variety developed by mass selection is made up of genotype bulk together without having undergone preliminary testing to determine their performance in hybrid combination. Synthetic varieties are known for their hybrid vigor and for their ability to produce usable seed for succeeding seasons. Because of these advantages, synthetic varieties have become increasingly favored in the growing of many species such as forage crops, in which expense prohibits the development or use of hybrid varieties. The asexual propagation methods are also there by which we can get the plants propagated. The major method of asexual propagations are cuttings, layering, division, budding, grafting, etc. The cutting involves rooting severed pieces of the parent plants. Layering involves rooting a part of the parent and then severing it, and budding and grafting is joining two plant parts from different varieties. Here we can see all these methods in diagram. The vegetative propagation methods. Here there is cutting, how cuttings are made, then mark coating, how wet fibers are used, and it is layering, process of asexual reproduction, and how budding and grafting is done here in the process. There are different methods of vegetative propagations are there by which we can multiply the plants. Many types of plants, both woody and herbaceous, are frequently propagated by cuttings. A cutting is a vegetative plant part which is severed from the parent plant in order to regenerate itself, thereby forming a whole new plant. Take cutting with a sharp blade or knife to reduce injury to the parent plant. Dip the cutting tool in rubbing alcohol or a mixture of 1 part bleach to 9 part water to prevent transmitting disease from infected plant parts to healthy ones. Remove flowers and flower buds to allow the cutting to use its energy and stored carbohydrates for root and shoot formation rather than fruit and seed production. Here the typical cutting has been shown in the diagram, the mother plant in which the cutting has been made, and this cutting is again planted in separate pot, and the new plant can be synthesized. Propagation by layering. Stem still attached to their parent plant may form roots where they touch a rooting medium. Severed from the parent plant, the rooted stem becomes a new plant. This method of vegetative propagation is called a layering. Promotes a high success rate because it prevents the water stress and carbohydrate shortage that plague cuttings. Some plants layer themselves naturally, but sometimes plant propagators assist the process. Layering may be enhanced by wounding one side of the stem or by bending it very sharply the rooting medium should always provide aeration and constant supply of the moisture to develop it into a plant. Here we can see the plant is shown here, as the branch of plant is buried into soil, and after some time we can see here roots are originated. Now this plant part above this layered part, we can see here the vigorous shoots are regenerated. So this part is removed and another new plant is synthesized by the layering. Propagation by grafting. Grafting is an advanced technique that botanists, farmers, gardeners and hobbyists use to add living tissue from one plant to the another. Plant tissues have the ability to grow new vascular tissues, and the process of grafting takes advantage of this ability. The vascular tissue of the main plants join with the vascular tissue of the added plant, and keeping the new tissue alive and growing. This is way for plants to reproduce asexually. You can think of grafting as being similar to the organ donation. When someone needs a new kidney or liver, they can get once donated. ग्राफ्टिंग से अर्थ हमारे यहाँ पर है कि दो प्लांट जिसके अंदर एक स्टॉक होता है, दूसरा सियोन होता है, और हम कह सकते हैं कि जो वैरायटी हमें ग्राफ्ट करनी है उसको काट करके और तैयार कर लिया जाता है, और दूसरा प्लांट जिसके अंदर हमें ग्राफ्ट करना होता है उन दोनों के वेस्कुलर टिश्यू को इस प्रकार से ज्वाइन करते हैं कि उनके अंदर एक कंटिन्यूटी का निर्माण हो जाता है, और कंटिन्यूटी का निर्माण होते ही उसके अंदर वेस्कुलर सिस्टम प्रारंभ हो जाता है, और इससे जो है जो हम हमारी इच्छा का जो ग्राफ्ट उस पर लगा रहे हैं वो एक नए पौधे के रूप में उसके ऊपर आगे बढ़ जाता है। यहाँ पर ग्राफ्टिंग दिखा रखी है, इस प्रकार से ग्राफ्टिंग करते हैं, और इस प्रकार से स्टॉक और सियोन जो है, स्टॉक प्लांट का होता है जिसके अंदर जो है उसको बेस्ट प्लांट के रूप में यूज किया जाता है, इसको हम स्टॉक कहते हैं, और सियोन जो कि हम तैयार करते हैं कि हमें किस पौधे का सियोन जो है उसके ऊपर फिट करना है। सियोन और स्टॉक को जोड़ दिया जाता है आपस में, और इस प्रकार से एक दूसरे को काटा जाता है कि दोनों एक दूसरे के अंदर फिट हो जाते हैं, और फिट होने के बाद में इसको एक रेप करके और इसको फिक्स कर दिया जाता है ताकि इसके अंदर कंटिन्यूटी बन जाए, और कंटिन्यूटी बनने के साथ ही पौधे की ग्रोथ प्रारंभ हो जाए। तो दो डिफरेंट प्लांट्स को एक साथ जोड़ करके उनके वेस्कुलर टिश्यूज़ को आपस में एक दूसरे से मिला करके और ग्राफ्ट तैयार करना पड़ता है, और इस ग्राफ्ट को तैयार करने के लिए दोनों ही जो प्लांट काम में लिए जाते हैं उनके स्तंभ की मोटाई लगभग एक जैसी होनी चाहिए ताकि जो है उन दोनों का वेस्कुलर टिश्यू एक दूसरे के साथ मैच करके एक लाइन के अंदर आगे बढ़ सके। Propagation by budding. Budding is the vegetative method of plant propagation and can be defined as an art of insertion of a single mature bud in it to the stem of the root stock in such a way that the union takes place and the combination continues to grow. It is grafting of a single individual bud instead of whole bud stick on scion as done in the case of grafting. There are several techniques of methods of insertion of bud in the rootstock. The adoption of any of the method like grafting depends upon the plant to be budded, situation, facilities and sources available. Here we can see the stock is made. Here eye has been created, and the bud which we have to plant is kept here in this stalk, and this eye later on acts as a bud which is going to give a new plant, and this process of putting bud in particular stalk plant is called as budding. So these are the methods for asexual propagation of plants. So here we have discussed all the methods of plant breeding, in which mass selection, pure line selection, plant introduction, acclimatization, hybrid vigor, and then mutation breeding, and finally the methods of asexual reproduction by which plant can be propagated. Thank you very much.